Great job, Kristen, and it's great to hear about the technologies that are being developed to help these critical cardiac patients. Up next, I'd like to welcome Dr. Besim Ogretmen to the stage to represent Lipo-Immuno Tech.
Good morning, everyone. I hope everybody can hear me. I'm Besim Ogretmen, in short, BES. It's a great pleasure to introduce our company, Lipo-Immuno Tech, this morning. Next, please. So our goal is really a therapeutic company, and we are trying to improve our cancer patients' lives with our two products, which are basically developed with our research programs at MUSC and Hollings Cancer Center. So maybe not many of you are, interested in lipids or biochemistry, but this is how we formed our company based on two important themes. One is the lipid research and the other one, immunology. So the company name was formed between these two collaborations in two themes. So our company, basically the logo, I drew this logo, but it is a very simple but very straightforward focus running for the cure for cancer therapeutics. Next, please. So we are two co-founders. So as I mentioned, my lab is working on the cancer lipid and cancer lipid metabolism. And our other co-founder is Dr. Shikhar Mehrotra. He's an expert in cancer immunotherapy. So our research labs are next to each other at the Cancer Center, Hollings Cancer Center. And frankly, I didn't know much about cancer immunology, and Shikhar didn't know much about lipids. But working together, we formed the company in 2018. And we have been working together with very nice collaborations. And now we have two interesting and very powerful products. Next, please. So we have two IP-protected products. One is the chemotherapeutic agent. In the first line is the LCL768. The names can be improved a little bit, I think. So it's a lipid analog drug that targets cancer mitochondria. And the second product is a rapid expansion formulation to expand these immune cells called T cells that can actually improve their cancer-killing abilities. And both of these products were developed either for our research grants from NIH or STTR funding, phase one funding from NIH as well. Next, please. So in the next few slides, I'd like to give you some information about these two products that we are focused on. So the first one, as I mentioned, is a cancer chemotherapeutics. It's a lipid analog drug that was developed in our lab. And it basically goes and attacks cancer mitochondria, which is, these mitochondria are the fuel source for energy for cancer cells. Next slide, please. So in this graphical summary, I like to a little bit give you information how it works. So this compound that we developed basically associates with the mitochondria in cancer cells. And then this accumulation in these cancer mitochondria removes the mitochondria from the cancer cell system by degradation, so the cancer cells don't have enough energy anymore. And so these products, basically this product came out from many mechanistic studies that we developed, and these are published in high-impact publications for mechanistic studies. And one example is on the right, published in one of the science journals. Next, please. So we did some important preclinical studies in mice using head and neck cancer tumor models. And as shown in the red squares, our product was very helpful to inhibit tumor growth in vivo. and we didn't see any toxicities to animals with this preclinical trials. So this is only one of the trials, so we have been completed a lot of important studies, and we are very hopeful that this will go on to toxicology pre-IND studies, and hopefully for the phase 1 clinical trials soon. Next, please. So our other product is the rapid expansion formulation. So this contains some advanced cocktails of antibodies, cytokines, and other molecules to improve this immune cell functions outside of the patient's body. So what we do is actually we take the immune cells from cancer patients and then put them through our formulation and technology so that we can improve the cancer patient's own immune cells to kill the tumors when we put the cells back to the patient. And so usually the cancer patients' immune cells are not very healthy, as shown in the upper slides. This is an EM picture of a T cell or immune cell. So with our technology at the bottom, the T cells become much more healthier and much more active for improving the anti-tumor functions. And this technology can also be used for expanding these chimeric antigen receptor or CAR T cells. So these cells also can be packaged and put through the patients as therapeutic agents. Next, please. So one more, please. So as I mentioned, the technology is very interesting and improved cancer treatment in the last few years. So this is immunotherapy-based technology, but it can be personalized for individual patients. So what we do, basically, we take the patient's own immune cells and then put them through our rapid expansion protocol so that we can get enough numbers of T cells outside of the body, and those could reach to about 900 million cells or so, and they become much healthier and more energetic. And when we put them back to the patient's own body, it works much better for anti-cancer functions. And as I mentioned, we can actually engineer these T cells using genetic manipulations also to target specific antigens on cancer's surface with using these CAR T cells. Next, please. So this technology also was developed with our many mechanistic studies, and one, I'll give you an example published in one of the Cell journals recently. Next, please. So this technology can be used for many different types of cancers. We use mostly melanomas, but in this figure, we also use these T cells for prostate cancer inhibition of growth in animals. And you can see in the red bar, our rapid expansion protocol expanded T cells work really well for these different types of cancers, including melanoma and prostate. Next, please. So more importantly, we are also looking for CAR T cell expansions, as I mentioned. So we are right now in the preclinical, we completed the preclinical trials. We are with the pre-IND negotiations with FDA right now. So this basically looking at the CAR T cells that are developed in our labs, and so we can expand them. And so this panel basically shows you how much on the left column, how much is the criteria would be, and on the right results column, how much expansion and purity we get through our formulation. And we exceed above and beyond what is FDA requires for the IND applications. So we are hopeful that this product will be used in clinical phase 1 trial for the treatment of patients with leukemias, hopefully as soon as possible once we get the clearance from FDA. Next slide, please. So we did some market analysis. So this is a very conservative analysis looking at head and neck cancers for chemotherapy and the immunotherapy for leukemia patients. So I think globally it's about $15 billion. But this is, like I said, very conservative because we have many other indications other than head and neck and leukemias. We also have other products that are on the pipeline coming up for the pre-IND studies as well. Next slide, please. So, we have a very straightforward exit strategy. So we like to complete phase 1 and phase 2 trials for treating patients with aggressive tumors for both of our compounds. And then hopefully we will be able to partnership with a large biopharma so that, these products can be FDA approved for patients' improvement of outcomes. Next, please. So in conclusion, we have 2 IP-protected products which seem to be very good cancer chemotherapeutic or immunotherapeutic agents or technologies. So basically, we are now seeking for partnerships to expand our phase 1 and phase 2 clinical trials so that we can go for FDA approval for both of these technologies. Next. So I will stop here and thank you for your attention. I'm happy to answer questions.
So did I understand correctly that you extract the T cells and do you genetically modify them? I heard there were 2 paths you could take or not. So which one is it?
So we can do both actually. We can take the T cells from patients and without any genetic manipulations, we put them through our formulation and put them back to patients. Or in some cases, there are some new technologies where you can actually target specific antigens on cancer surface. So those will be the CAR T cells that can require some genetic manipulation.
So would the path with FDA be easier if you do not genetically change them?
That's correct. But also these CAR T cells, there are some FDA-approved products already. So FDA is very familiar with this technology. Okay. So we are going for that. And also, that will be more profitable because you can actually sell individual CAR T cells after we expand them in our system.
Is this actually 2 platforms or 1 platform?
So we have 2 products.
So they're separate platforms?
Separate platforms. One is for chemotherapy, one is for immunotherapy.
Okay, and how long does the alteration of the gene take?
So for the CAR T cells, these are pretty optimized. So like, the one table I showed you for the pre-IND studies, it took us probably 3 to 4 months to get to this point. We could get about 920 million cells from the patient's T cells after the genetic manipulation.
Let me ask the question a different way. If I am a patient and I'm going to use your therapeutics, you draw cells from me, when will the therapy be back to?
Within a couple of months, we will get back the cells back to the patient.
Okay, and is there any collateral damage once the therapeutic is injected back into the body?
So that's our technology. Basically, what our technology does is that those cells that we put back, they persist much better than, their own patient's own T cells.
And one final question, and I'll use prostate cancer. Do I reach a point where my Gleason score is so high that this will not be effective?
Some cancers are not very responsive to immunotherapy, so prostate cancer is one of those. But looks like our technology, at least in the preclinical studies, was very effective for prostate cancer as well. So we have actually some medical oncologists in our board. So we, talk to them, and so they decide, which patients are best for this trial and which patients are best for others. So we collaborate with our medical oncologists to decide that.
And I'm sorry, one more question. The pre-IND stage, cost, it's expensive, isn't it?
You know, we are lucky that we have FRD at MUSC and Hollings Cancer Center, so we have a good partnership with academia and biotech companies. So MUSC covers some of that cost for pre-IND, application.
Along with that idea, could you give me an idea of just the time frame and resources required to move from this point to where it would be fully established as a practice, assuming things went well?
Yeah, so this pre-IND application is already ongoing. So hopefully as soon as they approve, so we have the products already, standardized. So we can get more than 90% of the immune cells expanded and about 920 million cells. And FDA only requires 200 million cells. So we are there. So we are actually starting to look into identifying patients with leukemias. Usually these patients don't respond to any existing technologies or therapeutics. So they are the ones that will be moved forward to this technology and this therapeutic. So we are working with Hollings Cancer Center oncologists to identify patients and going through this, hopefully within a few months. I'm saying maybe 6 months to be more conservative.
Do you need additional team members, additional laboratory space? How does that ramp up over time?
Yeah, so like I said, Joe, this is a very good partnership with us and the MUSC and Hollings Cancer Center. So we have to expand these cells, you need to have a clean cell facility. So these are GMP grade facilities that don't have any contamination or any other toxic molecules inside, so that we use MUSC's clean cell facility for expanding these cells using our technology.
Regarding regulatory approval, are they going to force you to focus on a single, cancer, or can you go through clinical trials with multiple indications at the same time?
Yeah, so since there is already approved, FDA-approved CAR T-cell technology for leukemias, that's why we are going for the leukemia, because that's already been approved. But for other indications, such as prostate cancer, which doesn't really respond to immunotherapy, so we need to show more preclinical studies that this technology works for those cancers like prostate. And so that requires its own pre-IND application for FDA.
You said it took 2 months before the cells could be returned to the patient after treatment. Isn't the disease progressing during that period of time, and isn't that a long period of time for those critically ill patients?
Yeah, that's a great point. So that's our idea is as soon as we can get to the patient, we, put back the immunotherapeutic agent. But working with, oncologists, so what they do is they put the patient on another therapeutic agent so that the patient don't just have nothing. So they use some standard therapeutic agent anyways for these patients until they become available for our cells.
One question, I think you kind of answered this, but I apologize if I missed it. Would this, could this act as an independent therapy, or is it always going to be in combination, and what would the combination be with other therapies?
Yeah, so it depends on the patient. So this is mostly the individualized therapeutics right now. So because we take our patients' own cells and then put them back. So some patients, based on their progress, either they require chemo and immunotherapy combination together, or some patients will respond better for initial individual therapy. So those are decisions that, the oncologists make, and we work with them, whatever is needed for this. But initially for pre-IND, you need to go for a single agent and then decide what next steps are. All right, thank you so much. I appreciate it.